Hey everybody, let's move on to mini lecture 13B, which is going to be dealing with smooth muscle. Remember, we have three types of muscle in our body. We have skeletal muscle, cardiac muscle, and smooth muscle. Now, skeletal muscle we dealt with in the previous mini lecture. Cardiac muscle we'll get to when we get to the cardiovascular system. So now this mini lecture is going to concentrate on smooth muscle. And smooth muscle has the slowest contraction and relaxation durations of all three types of muscle. It also has the lowest oxygen consumption rates, and it does require less energy to contract. It has the ability to generate force over longer periods of time when you look at all three types of muscle tissue. As a matter of fact, smooth muscle can generate its maximum tension with only a quarter of the cross bridges active, or with only about a quarter of the cross bridges actually formed. And smooth muscle can actually still generate a pretty significant level of tension even when it's overstretched. If you remember, skeletal muscle had an optimal length and it generated maximum tension when it was 80% of that length, or you could stretch it to another 20% and go 120% of its optimal length, and you would still get maximal tension. But if you went beyond that 127 point, that 120% of optimal length, the tension that skeletal muscle fibers could generate dropped off sharply. And if you went below the 80% optimal length, tension dropped off sharply. Well, smooth muscle is different. Smooth muscle actually can generate maximum tension when you go over those, go beyond that 80 to 120% range. It can be stretched quite a distance before you actually get to that optimal length. And this is important for the contractile ability of hollow organs and blood vessels, because you can't stretch an organ to its maximum, let's say maximum capacity, and lose the tension that the smooth muscle lining can generate. So that's something unique about smooth muscle. Now, what are the characteristics of smooth muscle? First of all, we're going to start off with learning that smooth muscle fibers have more variety. There's basically three kinds of skeletal muscle fibers, right? Fast twitch glycolytic, fast twitch glycolytic oxidative, slow twitch oxidative. Three types. Cardiac muscle fibers, basically, as far as I remember, there's only one type of cardiac muscle fiber. But smooth muscle fibers have a wide variety of characteristics with differing properties. So for example, the smooth muscle fibers you would find lining a blood vessel, the vascular ones, they're different than the smooth muscle fibers that line the gastrointestinal system, or those which are found in the urinary, the respiratory, the reproductive system. The smooth muscles that are found in our ciliary bodies of our eye are very different. So a single model of smooth muscle function is impossible. That being said, though, I am going to offer you probably the most well-described model of smooth muscle contraction. Just be aware that not every smooth muscle fiber may contract using that mechanism. The anatomy of a smooth muscle is very distinct. The smooth muscle fibers, instead of being arranged in parallel organization, are arranged in a lattice-like organizational pattern. So think of it kind of like a tennis net in which the fibers go in various directions. This is so that the contractile forces can be generated in multiple directions. With the parallel arrangement of skeletal muscle, they contract basically in one plane. Smooth muscle has to be able to contract in multiple planes. Also, if you think about it, there are numerous organs in our body that have multiple layers of smooth muscle. So added on to that, that gives basically our organs the ability to contract in multiple directions, often at the same time. Contraction of smooth muscle is controlled by hormones, just like skeletal muscle. So acetylcholine actually does play a role in the contraction of smooth muscle. However, there are other things like, did I just say acetylcholine was a hormone? I lied. I totally lied to you. So contraction is controlled by hormones, paracrines, and neurotransmitters. So again, neurotransmitters, acetylcholine. Smooth muscle and skeletal muscle both can respond to acetylcholine. However, adding on to this, with smooth muscle, you have the neurotransmitter norepinephrine. You have the hormone epinephrine. And there are other paracrine factors that can be secreted. So the take-home message of this is that smooth muscle responds not just to acetylcholine, but to a wide variety of chemicals, whereas skeletal muscle only responds to acetylcholine. Smooth muscle has a variable electrical properties. It's not always the hard and fast rule that you get an action potential and you get a muscle contraction or a muscle twitch. There are some smooth muscle that can hyperpolarize in response to an action potential and generate a twitch. There are smooth muscles that may not get up to a threshold potential and still generate muscle contraction, or what we call a twitch. Finally, on this slide, we see that multiple pathways also can influence contraction and relaxation. Some neuronal pathways will elicit contraction in a smooth muscle. Other neuronal pathways will cause will inhibit contraction of that smooth muscle. So smooth muscle actually is sort of an integrating center that gets neuronal signals from multiple neuronal pathways. It takes those messages from those neuronal pathways and integrates it and figures out what the heck to do. And that may be stimulating contraction, or that could be inhibiting contraction. Whereas when we go back to skeletal muscle, basically you have the somatic motor pathway, and if you activate that motor pathway, you will get skeletal muscle contraction. When you add in the other levels, the higher order neurons, that's where you can actually inhibit contraction. But that lower motor neuron comes out, and if you stimulate that lower motor neuron, you will get contraction. So that's kind of different as to what a smooth muscle does. A smooth muscle is the one that makes the decision as to whether or not it's going to contract or not. That decision-making capability is gone in skeletal muscle, and it's the neuronal pathway that determines whether you get contraction or not. A lot of these characteristics, and you notice I've contrasted them with skeletal muscle. I've created a table in your Canvas module on muscle physiology that actually compares and contrasts smooth muscle and skeletal muscle, and a lot of these characteristics are in that table. So I'd advise you to go and take a look at it. Smooth muscle contains 3 filaments, or what you could call 3 myofilaments: myosin, actin, and what are called intermediate-sized filaments, which don't participate in contraction. So this sounds like skeletal muscle, and you are correct. Both skeletal and smooth muscle have actin and myosin myofilaments. However, the forms of actin and myosin are specific to smooth muscle and skeletal muscle. So when you start talking about actin and smooth muscle, you have to say smooth muscle actin or smooth muscle myosin. When you refer to actin and myosin in skeletal muscle, you should say skeletal muscle actin, skeletal muscle myosin. That's because these proteins are slightly different in smooth muscle versus skeletal muscle. For example, smooth muscle myosin and actin myofilaments are longer than their isoforms in skeletal muscle. There's more actin in smooth muscle than skeletal muscle fibers. So there's a little bit of difference in those myofilaments. In terms of the regulatory proteins, here's something really important. Smooth muscle do not have troponin. They only express tropomyosin. And it's not going to be involved in regulation of cross-bridging. So the cross-bridging in the model of smooth muscle contraction that I'm going to show you has got nothing to do with tropomyosin. One last thing on this slide that I'm going to point out to you is that there is no T-tubule structure in smooth muscle, and there is a very poorly developed sarcoplasmic reticulum. So the neural excitation within the excitation and generation of an action potential coming from the neurons is going to be a little different in a smooth muscle. We're going to see, yes, that neurons can stimulate smooth muscles to contract. We're going to see that an action potential can form in smooth muscle and that it will travel along the sarcolemma. But beyond that, it's going to be really, really different versus skeletal muscle cells. Smooth muscle fibers have no sarcomeric structure. There are no Z lines, there are no parallel arrangement of thick and thin myofilaments, so that sarcomere structure that you learned, toss it out with smooth muscle. It doesn't exist. Instead, what you have is a lattice-like pattern in which the fibers run in a 3-dimensional sort of network. The actin filaments attach to the sarcolemma through something called a dense body. And then the actin filaments will form a 3-dimensional network, and then the myosin filaments will form a 3-dimensional network in among them. Now, these dense bodies have the same proteins as Z lines, but they are just not found as parallel lines with the sarcomere in between. So the way I do it is, let me draw you this silly diagram. So I have basically, this is our sarcolemma. I'm going to draw a dense body here. I'm going to put a dense body here, maybe put one in the middle. So these are our DBs. They are made of the same proteins, the same kind of connectins that you would find in the Z line of a skeletal muscle. To this, I'm going to add actin filaments. Let's put them this way. So these are our actin thin myofilaments. And then I'm going to draw the myosin kind of like this. So these are our myosin filaments. So you can see these suckers do not run parallel to each other. They run in all kinds of directions. Maybe add another dense body here, put an actin filament going that way. Add a dense body here. Oops, sorry, that should be blue, right? Put a dense body here, put an actin filament this way. Put a myosin filament this way, put a myosin filament that way, a myosin filament this way. So can you see that there's absolutely no parallel organization? That is the lattice-like organization that I described earlier. And of course, this picture on the right-hand side shows it a little bit neater, but this sort of three-dimensional network of interactions between thick and thin myofilaments means that the smooth muscle can contract in multiple directions, often at the same time. Let's learn a typical model mechanism for smooth muscle contraction. Again, I'm going to reiterate that there is no one model that summarizes contraction in every kind of smooth muscle fiber. But I'm going to show you the most well-described and well-studied model. And it starts off with an action potential flowing along the sarcolemma. So there's our action potential. It's going to start and it's going to flow along the sarcolemma. Now, this action potential can be generated in response to neuronal stimulation, or it can actually be generated spontaneously. Either way, action potential travels along. It is going to cause the opening of voltage-sensitive calcium channels. So these are going to be voltage-gated calcium channels that are going to be embedded in the membrane. Calcium is now going to flow into the smooth muscle from the extracellular environment. So we are going to get a small but definite increase in calcium levels within the smooth muscle fiber. This increased intracellular calcium is actually going to open calcium release channels that are found in the sarcoplasmic reticulum. These are the same ryanodine foot calcium receptors that you will find in the sarcoplasmic reticulum of skeletal muscle. So this slight increase in intracellular calcium, which has come in from the extracellular fluid, is now going to trigger the opening of calcium release channels in the sarcoplasmic reticulum. That calcium is going to come out. And now we have, again, a very large increase in intracellular calcium. And then that calcium can then be used in the subsequent events of contraction. This is called calcium-induced calcium release. And I want you to stick a pin in this because this is going to be the similar mechanism where we will eventually see in cardiac muscle. The overall effect is you have a pretty significant increase in intracellular calcium levels following that action potential. The regulation of contraction in smooth muscle does play a role, does rely upon calcium. The actual mechanism is calcium-dependent phosphorylation of myosin heads. Now, since there's no troponin, how can a smooth muscle cell regulate cross-bridging between actin and myosin? It has to do actually with calcium and phosphorylation. The answer to this comes from the fact that myosin, remember, myosin is not only a golf club made up of a shaft, a hinge, and a head, but that's the myosin heavy chain. Myosin also contains light chains. Now, I'm going to draw the light chain kind of like this across the head of the myosin heavy chain. And the reason why I draw it is because the myosin light chain covers up the actin binding site. So in a way, the myosin light chain plays the role of tropomyosin. If you increase intracellular calcium, you will end up with a series of biochemical events that is going to phosphorylate this myosin light chain, change its shape, and shift it off of the actin binding sites that are found on the myosin head. Now, the difference between this and skeletal muscle is calcium directly shifts off the tropomyosin from the actin binding site. In smooth muscle, calcium is going to lead to a series of events that's going to phosphorylate the myosin light chain, causing the myosin light chain to come off the actin binding site. So let's see how that works. Remember, ultimately the action potential is going to cause an increase in intracellular calcium. That increase in intracellular calcium primarily comes from the sarcoplasmic reticulum, but that increased intracellular calcium now is going to cause the activation of a kinase called calmodulin. Calcium is going to bind that protein called calmodulin, turn it on. Calmodulin is then going to find its target, phosphorylate and activate it. It is called a kinase that is oddly enough called myosin light chain kinase, or MLCK. Myosin light chain kinase is going to get phosphorylated and activated, and it's going to find its target. And its target happens to be the light chains found on the head of myosin. So it is going to essentially phosphorylate the myosin light chains, changing their shape, pulling them off of the myosin head and exposing the actin binding site on the myosin head. Now the myosin can cross bridge with the actin. So this is calcium-dependent phosphorylation of the myosin light chain, causing it to shift its conformation off of the actin binding site, and now you can get cross-bridging. The phosphorylation also will increase the ATPase activity of the myosin head as well. So not only do we expose the actin binding site on the myosin head, but we increase the ATPase activity. So again, if you were to just draw the myosin head here, I'm going to put an actin binding site right there. There's our ABS, actin binding site. I will put an ATPase site right there. ATPase site right there. Our myosin light chain right there normally covers that up. Phosphorylate it, it will shift it off. The actin binding site is exposed, and the phosphorylation also activates that ATPase site. You guys know that the somatic motor pathways control skeletal muscle contraction. So we've learned that skeletal muscle doesn't contract unless a motor neuron tells it to. That's not necessarily the case for smooth muscle. So to finish up now, let's talk about what is known as multi-unit and single-unit smooth muscles. And this ties into how these smooth muscle cells generate their action potential. We're going to start off with multi-unit smooth muscle. The fibers exhibit neural-like properties. The muscle fibers within the smooth muscle contract as a whole as one unit. The cells are not linked to each other electrically. Okay, so in order for all of these smooth muscle fibers to contract at once, each muscle cell must be closely associated with an axon terminal. And the autonomic neurons that innervate multi-unit smooth muscle are weird. They don't end as the typical axon terminals with, these little synaptic vesicles. They don't do that. They actually have these really long terminals with synaptic end bulbs all along the end. So they're more like this with these little beads attached, kind of like beads on a string, where each of these beads represents a synaptic end bulb storing action potentials. Storing neurotransmitters. So you take these beads and you weave them in and around all of the smooth muscle fibers. And again, remember, each of these little beads is a site for the storage of a neurotransmitter. So when you stimulate these neurons and they release their neurotransmitters, the neurotransmitters can then bind and stimulate the contraction of the smooth muscle. This kind of very intimate association with multiple synaptic end bulbs and multiple smooth muscle fibers allows for fine control of contraction. Multi-unit smooth muscle are found in things like the iris and the ciliary body, parts of the male reproductive tract, and in the uterus. Although in the uterus, the smooth muscle will shift to single unit as pregnancy proceeds. But this gives very, very fine control, because if these synaptic end bulbs release their neurotransmitters, the nearby smooth muscle cells will contract. But if these ones over here at the opposite end do not release their neurotransmitters, then the nearby smooth muscle cells that are supplied over at that area will not contract. Gives rise to very fine control of a smooth muscle. Okay, remember, multi-unit. You've got this network of autonomic nervous system neurons, which kind of look again like beads on a string. They weave their way in and around the smooth muscle so that multiple smooth muscle cells will contract as a unit when a group of synaptic vesicles release their neurotransmitters. All right, now in single unit, you can have the contraction of the muscle as a separate single unit. So single unit smooth muscle is also called myogenic smooth muscle because contraction originates from the property of the muscle cell itself. This is the kind of smooth muscle you will find lining the walls of organs and blood vessels. As a matter of fact, most of the smooth muscle in our body is single unit. In this picture, you can see this really weird autonomic neuron with, again, its long terminals with sort of these bead-like synaptic vesicles. In single unit, let's say that this synaptic vesicle releases its neurotransmitter and the nearby smooth muscle fiber generates an action potential. Each one of these smooth muscle fibers are connected to each other with intercellular junctions called gap junctions. So now you have the action potential spreading throughout this entire network, and the entire muscle contracts as a result. Not a group of muscles which contract as a unit, like in multi-unit, but the entire muscle contracts. The amount of calcium that enters a single unit smooth muscle can also determine the force of contraction. Remember, the more intercellular calcium you can increase in a smooth muscle fiber, the more of those kinases will get activated, the more myosin light chains will get phosphorylated, the more cross-bridging you can have. And that happens in smooth muscle single unit cells. So again, I think I have a table that compares and contrasts single unit and multi-unit in the Canvas shell. Go take a look at that because it's important that you understand the difference between a section of a smooth muscle contracting as a unit, that's multi-unit, or the entire smooth muscle contracting as a unit, that's single unit. To help, I found again a couple pictures. I took the multi and single unit figures we see in our previous slides, put them together. So you can see that intimate connection between the autonomic nervous system and the independent multi-unit smooth muscle fibers, as opposed to the lower innervation and the interconnectedness of single unit smooth muscle. And then on the left-hand side, I found another picture. Again, in multi-unit smooth muscle, each smooth muscle fiber has to have some kind of connection to the autonomic nervous system. And in smooth muscle, because all of these smooth muscles the cells are physically connected to each other, you don't need as much nervous innervation. What's really interesting about single unit smooth muscle is that they don't necessarily have to have neural stimulation. The smooth muscle cells of single unit smooth muscle can generate their own action potentials spontaneously. They do not need neural stimulation. Multi-unit smooth muscle does. So single unit smooth muscle does not. As a matter of fact, in single unit smooth muscle, there are specific smooth muscle cells that are specialized to initiate action potentials, but don't necessarily contract. And the reason why these cells are specialized to initiate their own action potentials without the nervous system is because their resting membrane potential is unstable. It will fluctuate automatically without any external influence. They will spontaneously begin to depolarize towards threshold. Now, these types of smooth muscle cells can be either a pacemaker or slow wave. In the pacemaker, you're going to have a regular and rhythmic spontaneous depolarization towards threshold so that these smooth muscle cells regularly and rhythmically generate their own action potential on a regular rhythmic basis. And in slow wave potentials, you're going to see that there's not necessarily a rhythmic pattern. In slow wave, they do exhibit cyclic depolarization and repolarization. However, they might not cyclically reach threshold and they might not cyclically contract. So while depolarization and repolarization might appear to have some kind of rhythmic cycle, the reaching of threshold and the generation of an action potential and the contraction does not follow any kind of rhythmic cycle. This is in contrast to pacemaker. Pacemaker smooth muscle cells will always reach their threshold potential, and they will always generate an action potential and contract on a regular, cyclical, rhythmic basis. Single unit smooth muscle interconnection ensures that the entire muscle contracts upon the initiation of an action potential. Remember, single unit smooth muscle contracts as a unit because they're all connected to one another electrically through gap junctions. So when an action potential starts in one smooth muscle cell, it will spread through all of the other interconnected ones. This means, though, that the smooth muscle can't vary the number of muscle cells contracting because they're all interconnected with one another. But it can vary the tension of contraction by changing the level of calcium found in the sarcoplasm. By varying the amount of calcium in the sarcoplasm, the cytosol of a smooth muscle cell, that can alter the number of eventual cross bridges that will form, and that can eventually alter the strength of smooth muscle contraction. As a matter of fact, many single unit smooth muscle cells have sufficient calcium within their cytosol to ensure a low level of constant contraction, and this is referred to as smooth muscle tone. Remember that smooth muscle is innervated by the autonomic nervous system, and the two neurotransmitters of the autonomic nervous system are acetylcholine and norepinephrine. Now, these neurotransmitters do not initiate smooth muscle contraction. Remember, that's done by an action potential and by calcium levels in the sarcoplasm. But these neurotransmitters can modify the rate and the strength of contraction in single unit smooth muscle. Smooth muscle activity can also be modified by things like hormones, muscle stretch, and by certain drugs. And these neurotransmitters and things like hormones, muscle stretch, and drugs, they all act by modifying the permeability of the sarcolemma to extracellular calcium. There's a compound that's made by endothelial cells that line a blood vessel and also by neurons. It's called nitric oxide. Don't get this mixed up with nitrous oxide, which is laughing gas. Nitric oxide is produced by certain cells in the human body, specifically endothelial cells and by neurons. Nitric oxide acts to inhibit smooth muscle contraction, and it can induce smooth muscle relaxation. And it does so by stimulating the production of a second messenger in smooth muscle cells. And as a result of that production of second messenger, the blood vessels will dilate as the smooth muscles lining them relax. Okay, so why am I telling you about nitric oxide? Well, I want to give you a little bit of a medical application here, which is going to be how nitric oxide, smooth muscle, and the drug called Viagra interact with one another to basically treat a condition known as erectile dysfunction. The smooth muscle cells that line the blood vessels supplying penile erectile tissue, they make a second messenger called cyclic GMP. CGMP. And if this sounds familiar because there's kind of a cousin or a relative to cyclic GMP that we've talked about already, it's called cyclic AMP. These second messengers work basically in similar ways, that they cause specific intracellular events to happen. Now, cyclic GMP is made by an effector protein called guanylate cyclase. Guanylate cyclase takes GTP and converts it to cyclic GMP. And again, this should sound familiar because the effector called adenylate cyclase converts ATP to cyclic AMP. So again, lots of commonalities. Guanylate cyclase activity is stimulated when neurons release nitric oxide. The nitric oxide stimulates guanylate cyclase activity to basically dramatically increase, and you get a whole bunch of cyclic GMP made inside the smooth muscle cells that line the blood vessels supplying penile erectile tissue. There's a whole bunch of things that happen inside that smooth muscle cell, but ultimately calcium levels are going to drop, and that means you don't get as much smooth muscle contraction. So the end result of nitric oxide being stimulated is that inside the smooth muscle cell, cyclic GMP rises and the smooth muscle cells don't contract, instead they relax. This leads to vasodilation of the blood vessels leading into the erectile tissue. More blood flows into the erectile tissue and you get an erection. However, cyclic GMP is broken down over time by an enzyme called
Cyclic GMP phosphodiesterase 5. That's yike. Anyway, we're going to short form this and we're going to call it PDE5. This is an automatic mechanism that is meant basically to eliminate the erection. So over time, cyclic GMP is going to be broken down in these smooth muscle cells by an enzyme called PDE5. The relaxation of the arterial smooth muscle will gradually start to disappear, and eventually the blood flow will leave the erectile tissue and the erection will disappear. Now the question is, how does Viagra mess with all that? Well, click on over to the next slide and I'll tell you. Viagra, which is known as sildenafil, inhibits the activity of this PDE5 enzyme, and that's showing it right here. This PDE5 enzyme basically is knocked out. This means that the gradual breakdown of cyclic GMP is going to slow down in smooth muscle cells. And if you lower this PDE5 activity, you're going to decrease the cyclic GMP breakdown. You've basically got, you're going to maintain those high levels of cyclic GMP. That's going to lower eventually calcium levels inside the smooth muscle. The arterial smooth muscle is going to relax. Vasodilation is going to be maintained, and the erection is going to be maintained over time. So the idea is cyclic GMP relaxes the smooth muscles that line these blood vessels. You get more vasodilation. More cyclic GMP means more dilated blood vessels. More vasodilation means more erectile tissue filled with blood and a longer erection. Funny story about Viagra, though. This was not actually discovered and researched as a drug to treat erectile dysfunction. It was actually first proposed to treat strokes and heart attacks. Because it causes relaxation of arterial smooth muscle, they thought this could be applied to the coronary muscles supplying heart muscle. They discovered that it did not work. It failed miserably as a drug to treat coronary heart disease, but they did notice a very suspicious and common side effect in men. And voila, Viagra was born.